Okay, so we've talked about problems when the chromosomes do not separate appropriately during meiosis, and now we're going to talk about specific chromosome structure and arrangement problems. There are a couple of different types you can get. You can have deletions where one or more chunks of the chromosome is lost. Duplication, where you have a presence of a chromosome segment more than once. Inversion, where part is lost, part is broken and it's replaced but in the backwards direction. And a translocation, where it might break off from one chromosome and then move to another chromosome. And this can be either reciprocal or non-reciprocal. So let's look visually at what's happening here. In a deletion, you might have a chromosome here, A, B, C, D, E, F, G. Okay, but there might be a deletion where you have a breakage and a loss of, right here would be section D. So you would go A, B, C, E, F, G. This is probably arguably more common at the ends of the chromosomes, but it certainly can happen quite frequently within the center of the chromosomes. And we'll talk a little bit more about that in just a minute. You can also have a duplication. In this case, it's going to be duplication of region BC. So if you look here, A, B, C, D, E, F, G. Okay, and then it goes, after the duplication, you would have A, B, C, B, C, D, E, F, G. So you would have this BC region twice in there. In an inversion, again, it's broken and then replaced, but in the backwards direction. So instead of A, B, C, D, E, F, G, you would have A, D, C, B, because that's backwards, E, F, G. In terms of translocations, it can be either non-reciprocal or reciprocal. So if something is going to be non-reciprocal, what's going to happen is part's going to break off, it's going to go and attach to another chromosome, but that other chromosome isn't going to give anything back to it. And so here you would have two different chromosomes, A, B, C, D, E, F, G, a separate one, H, I, J, K, L, M. The A and B might break off and go and attach to the end of this H, I, J, K, L, M chromosome. So now you have still two chromosomes. One, though, is going to be shorter than it's supposed to be. One is going to be longer than it's supposed to be. In a reciprocal translocation, you would have part of this chromosome break off, part of that chromosome break off, and they're going to swap with one another. So in this case, the HIJ is going to break off, and it's going to attach to this CDEFG. The other chromosome, the A and the B, is going to break off, and it's going to go and attach to the KLM. Let's look a little bit closer at a few of these examples. So in terms of deletions, there's two main ways that you're going to be able to get a deletion. The first is if you're going to have a break somewhere, and that little section that got broken off is just lost. The second is if you have these little twists that can form. In this case, you have A, B, C, D, and then since it's got all twisted, maybe these two parts broke off, okay, and they can be lost. These two parts are then going to reseal next to each other. So a case study for this is Cri-du-Chat syndrome. This is a deletion of a small terminal portion of chromosome 5. So the genetic designation for this is 46 because it still has 46 chromosomes, 5p minus. So remember we talked about the p arm, the q arm. What's happening here is a small portion of the p arm of chromosome 5 is lost. So this is a variable syndrome. The more that's deleted, the greater the impact on the children who survive. So if you only lose a very small portion of the chromosome, you're not going to have as severe of a problem as somebody who loses a much larger section of their chromosome. Some of the symptoms of this disorder: patients have an eerie cry, and it sounds similar to a meowing cat, and that's actually how it's got its name, because that Cri-du-Chat means is the cry of the cat in French, due to abnormal development of the glottis and the larynx. You also might have a patient with gastrointestinal and cardiac complications, so it's not just this weird cry. They really do have significant developmental problems. And it's estimated approximately about 1 in maybe 25,000 or 50,000 live births. And so this is what the genetic profile of an individual with this disorder. You can see here chromosome 5, here is intact chromosome 5, here's the second chromosome 5, but it happens to be missing a chunk at the end here. So duplication, one of the ways that you can get duplication is due to unequal crossing over. So say you have some crossing over happening, chromosome 1, this one's not being crossed over, but here 2 and 3 are crossing over, okay? They're going to be exchanging bits of genetic information, but it's not happening equally. So what you're going to have here, instead of having 4 chromosomes that all have an A, B, C, D, E, F, you're going to have, this one's going to have A, B, C, D, E, F, but this one's only going to have A, B, D, E, F. This one's going to have that extra C segment here, D, E, F. And then this chromosome would be fine as well. So we have copy number variants, or CNVs, are going to be deletions and duplications at the molecular level. So rather than just huge chunks that we're talking about, we're talking about maybe about 1 kb in length. This is a typo here, it should be kb. It's found in coding and non-coding regions of the genome, so both within and within genes as well as in the non-coding regions of the genome. We define them as regions of DNA that have at least 1 kb in length or 1,000 base pairs in length that happen to display at least 90% sequence identity to one another. So there's been some ongoing studies trying to identify them in humans, like how many is normal for different genes or different regions, and they found that over 10,000 different regions have been identified in humans. And so studies are ongoing to try to associate them with human diseases. And they found both positive and negative associations with diseases. So they found that, for example, about 10% of the sporadic autism cases happen to have a mutant CNV site. In contrast, there can also be positive associations with diseases. So there are some mutant CNV sites that are associated with better outcomes from certain cancer treatments. And so their studies are ongoing to really just try to catalog and associate them, all the known ones, with human diseases. Okay, so inversions are going to require 2 breaks in the chromosome. So what happens here is you're going to have maybe part of a chromosome twisting up like this. You're going to have a break, some gaps created, and it's going to reseal, but instead of resealing back to where it should be, A to B and E to F, A and E are going to combine, and the F and B are going to rejoin instead. And so the end result of this is going to be an inverted sequence. So instead of the A, B, C, D, E, F, A, E, D, C, B, F. And this part right here is just happens to be inverted. So translocations, again, are going to be the movement of the chromosome segments to a new location in the genome. If it's reciprocal, the exchange between the 2 non-homologous chromosomes. The genetic consequences of this is going to be similar to inversions. You don't have any genetic gain or loss of information, just a rearrangement. If something is non-reciprocal, some of these are also known as a Robertsonian translocation. What happens is you have a break at a short arm of a chromosome. That small part is lost, and the large part is fused to another chromosome. So in this case, you do have a genetic loss of information, just like you would with a deletion. The difference is the remaining chromosome, instead of just being on its own, it's going to attach to part of a different chromosome. So there are cases where it's known that this translocation is involved with familial Down syndrome or inherited Down syndrome. So most of the cases of Down syndrome do not run in families, but there are a few cases, we call it familial Down syndrome, where it does run in families. And so what happens here, let's look at a normal patient, a normal gamete. You have chromosome 21, chromosome 14. After meiosis, you're going to have 1 copy of each of these. Now this carrier, what's going to happen? You have chromosome 21, but the other copy of this chromosome 21 gets attached to, in this case, chromosome 14. So what's going to happen during this gamete formation, here you're going to have this somewhat normal gamete. Here it's going to be this weirdly fused one. Here it's going to be part normal, part fused, and in this case it's going to be just this chromosome 14. So the resulting gametes from this, okay, if it combines with this normal gamete here, you might have a normal offspring. So you do have a chance of having a normal offspring. You may have a translocation carrier. You may have an individual with Down syndrome, and this is because there's 3 copies technically of chromosome 21, even though this copy happens to be stuck to chromosome 14. You can also have potentially this monosomic, which would be lethal for the individual. And what this one would look like here, you can see this chromosome 21, here's copy 1, copy 2, copy 3, which is stuck onto the chromosome 14. Another case study, Fragile X syndrome, also known as Martin-Bell syndrome. It is the most common form of inherited mental retardation. These individuals are very sensitive to a lack of folate, particularly during development. It occurs in approximately 1 in 4,000 males and 1 in 8,000 females. Now, the reason for this is it's a dominant trait on the X chromosome, but the penetrance of this is not complete. And we'll talk more about what that means in the Chapter 3 material. But basically what it means is it's going to be a dominant trait, but it's just not 100% dominant. Okay, because it's a little bit leaky. So the end result is that the trait is fully expressed in about 30% of the Fragile X-bearing females and about 80% of the Fragile X-bearing males. And that's why you're going to see these differences in the numbers here. So these fragile sites, there's also a link between many of these fragile sites on the chromosomes and cancer. So when I'm talking about fragile sites, I'm talking about sites on the chromosome that are very susceptible to breakage. And what they found is that genes within these regions have increased susceptibility to mutations and deletions. So one of the known ones, they call it FRA3b. It's a fragile site on the P arm of chromosome 3. That's where it gets its name from. This gene is often altered or missing in cells taken from individuals with lung cancer. They've done some further studies. They've revealed that normal protein product of this gene is absent in cells of many other cancers as well.